Thank you for having us. So today we're going to sort of split our talk and sort of talk about our new work in sort of expanding the CRISPR world and the types of tools that we can use for human health. And two things that we'll particularly talk about are RNA editing instead of DNA editing and sort of new areas in CRISPR diagnostics. So to dive right in, Cas9 is really exciting because it's really enabled all these new applications in genome editing. But when you look at where Cas9 is in the CRISPR universe, it's actually really just a small part of what's going on in CRISPR and bacteria. And so a question we really had a few years ago was, could we computationally mine bacterial sort of gene systems to find new types of CRISPR enzymes that could be useful for new types of tools? And so Cas9 is really exciting because it allows for programmable DNA cleavage, which can then be used for genome editing. But through our computational work, we actually discovered an enzyme Cas13 that could target RNA instead. And RNA is really interesting because it's temporary in the cell. And so if you do RNA editing or RNA perturbations, they can always be reverted if something bad happens. And off-targets are a lot less of an issue. So RNA is very exciting for us. And what's cool about Cas13 is it also allows for programmable RNA cleavage. And it also leads to a very special effect called the collateral effect, which is where when it recognizes a target, it'll just cleave any RNAs in a cell or solution. And Jonathan will actually talk about how we can leverage that for really exciting CRISPR diagnostics. And so to dive right in, right now I'll talk about RNA editing and how that's useful therapeutically, and then we'll talk about diagnostics as well. So in the cell, there are these really cool enzymes called ADARs, which can do precise adenosine to inosine changes. And inosines will actually be read out as a guanosine, so what you can do is do very precise A to G changes. And so that's useful because there's a lot of adenosines in the cell that can cause disease. Last time I think we looked, there's like 25,000 diseases that could be sort of corrected if you could convert an adenosine to a guanosine. And the way we can do precise A to G editing is we take sort of Cas13, which is here, and we can fuse this ADAR enzyme to it. And using the guide RNA, we can actually create the favored substrate of the adenosine deaminase, which will then recognize that region where you target and create an A to G change very precisely. And that's useful, and we, of course, gave it an acronym called REPAIR, which sort of is kind of a backronym for the process. So this is useful because there are many, like I said, diseases like, for example, nephrogenic diabetes that could be corrected from an efficient and precise A to G change. There's the multiplexing potential of the system. So CRISPR enzymes can really do multiple edits very efficiently. So in diseases where there are multiple edits that you might want to make, we can do it very well. You can also engineer pathways this way. We can also affect splicing by changing splicing mutations. And so you can actually change the way information is conveyed in the cell. And lastly, we can really modulate protein function temporarily. So you can sort of change phosphorylation sites or other sites in the sort of enzyme core to affect protein function. And so we're really excited about the thousands of genetic diseases that we might be able to correct with a technology like this one day, including many neurodegenerative diseases, neurodevelopmental diseases, and sort of we're going forward now and trying to show how RNA editing could be used sort of in mouse models. And so we really envision one day maybe there could be a pill for gene therapy where when you take it, you're actually modulating your own genetics. If something bad happens, you could stop taking it. We really get around a lot of the issues that we've been discussing about, is permanence bad? And maybe RNA editing could get around that. So, yeah.
Awesome. So I'll talk a little bit about how we can actually use CRISPR not to edit or change DNA or RNA, but actually to detect nucleic acids. So this is one thing that we never thought that we'd be able to do, use CRISPR as a diagnostic, but it actually is a very powerful tool as well. So this really relies on this collateral activity that Omar talked about, where our enzyme, if it finds the piece of RNA or DNA that we're targeting it to, will actually become activated. And we can use that activation as an indication that a nucleic acid is present or absent. So when it becomes activated, we can dope in something like a reporter that when cut apart, it will actually be able to glow or have another indication that there's a presence of something there. So that turns us into a detection technology, and because it's detection, we went with Sherlock. But it allows it to have a very specific and sensitive detection of nucleic acids. Because the CRISPR enzymes are very specific, we can detect single nucleic acids, and because it's very sensitive, we can detect very small amounts of analytes. So we've used this for many different applications. We can detect viruses such as Zika or dengue. We can detect different types of bacterial isolates as well as antibiotic resistance genes. It can be freeze-dried for point-of-care detection and requires no temperature changes. So we can run it with minimal and in some cases no instrumentation at all. You can use it to sense single base pair changes to genotype patients directly from saliva sometimes. And lastly, we can actually use it to detect cancer-related mutations in circulating DNA. So if you have a tumor, you'll shed mutations into your blood, and we can actually take that blood and detect mutations directly. So the power of this is that it's very portable, and it can be used in many different readout possibilities. So one of the most exciting readouts we've done is actually a lateral flow strip, which you might be familiar with the technology in things like pregnancy tests. So this is actually a programmable readout of different nucleic acids, and we can code them for, in this example, either Zika or dengue, and you can see that top band develop when the sample is present. So we've been working with collaborators where we can take samples directly from urine and detect Zika within an hour with essentially no instrumentation at all. So we're very excited to take this technology. As we know from the New York Times, DNA is all around us, and when we can sense it, it makes it very powerful for human pathogens in agriculture. So in general, DNA down here is a very robust toolbox now with CRISPR tools, and you've seen and will see throughout the next day the potential. And we're very excited about how we can fashion the RNA toolbox and generate tools there that we can use for human health with either RNA editing, nucleic acid detection, or many other things. So with that, we'd like to thank the people who made this possible, most notably Feng, who you may be hearing about quite soon, but also David Cox, who was a team member with us on the RNA editing work, and all the members of the Zhang Lab who helped make this possible. So thank you.